Welcome to our first lecture in microbiology. This lecture is entitled Introduction to Microbiology, and it will cover the three sections of Chapter 1 listed here: what our ancestors knew, a systematic approach, and types of microorganisms. So let's begin by first defining what a microorganism is. It is basically a very small organism that's too small to be seen without the use of a microscope. Now, throughout the semester, you're going to find that microbiology will also include the study of some organisms that can be seen without a microscope. The reason why we do include them is because they are genetically related to these small microorganisms. Now, of all the identified microorganisms, or the microorganisms that we know of today, only 1% of them act as pathogens. That means that these pathogens have the ability to cause disease in healthy individuals, whereas the remaining 99% are non-pathogenic or not a pathogen. For the most part, our existence is highly dependent on the presence of these 99% non-pathogenic organisms. Without them, life on Earth would cease to exist. So during the semester, I'll try to impress on you the benefits as well as the harm that's caused by these different microbes. But for now, let's take a look at how the study of microbiology came to be. Before microbiology became a formal study of science, our ancestors actually benefited from microbes without even knowing it. Now, I'm going to say microbes sometimes because it's the short way of calling a microorganism, and sometimes easier to pronounce. So, for an example, a lot of the food products that were made by our ancestors were made with the aid or the use of microbes. For example, yeast, which is a fungi, has been used for over 4,000 years to make bread rise. Before that, our ancestors had or would make unleavened bread. So we need yeast to make bread rise, and we call it leavened bread. Now, we know that they used a form of yeast because they had early reference of a practice that was found in the Egyptian writings. In that time, they referred to the bread as barn, B-A-R-N. Now, we know that these Egyptians accidentally included the source of yeast that fermented sugars in their wheat and produced carbon dioxide that then ultimately made the dough to rise. Today, we know that microorganisms are the cause of this, and we know that it's baker's yeast. So we can go to the store and get a package of Fleischmann's baker's yeast, which is also Saccharomyces cerevisiae. At about the same time, we found a Mesopotamian poem that's written to pay homage to the goddess of brewing. Her name is Ninkasi. Along with this poem were ancient beer recipes. Today we know that again it's yeast that are used to ferment the grains or fruits sugars to produce ethanol found in beer, wine, and other alcoholic beverages. Now, besides producing food products, certain microbes can cause disease, as I have inferred to earlier. These diseases can be found in humans and other organisms. But before the link between pathogens and infectious diseases were made, our ancestors thought that diseases were a form of punishment that was handed down by their gods for certain misdeeds. Over time, experts tried explaining the cause of these diseases as something that was more tangible, such as the bad air. Because crowded, poor sanitary conditions was often associated with bad odors, and such living conditions often led to the rapid spread of disease, people associated foul odors as a cause of disease. Thus, the miasma theory was born. This theory states that diseases are caused by a noxious form of bad air. So up to this point, microbes were not discovered. The actual discovery of microbes had to wait for the invention of a microscope that was powerful enough to see microscopic life forms. Although Anton van Leeuwenhoek wasn't the first to invent a microscope, his microscope was actually the first one that was able to magnify images big enough to actually see bacteria and protozoas. Now, the story has it was that van Leeuwenhoek was a Dutch merchant who bought and sold linen. He made a single lens handheld microscope shown here so that he could examine the threads of his linen. The idea was that the better the quality of his linen, the more money he could get for them. Now, van Leeuwenhoek's curiosity led him to more than just looking at his linen. He also looked at drops of water, human samples, and other biological samples. What he noticed was that they had some small objects that appeared to be moving inside these samples. He called them animalcules, which today we know them as being bacteria and protists. The image on the right is actually a sample of his drawings that he had published in a letter to the London's Royal Society in September of 1683. Now, the discovery of these small animalcules, these single-cell organisms, opened up many questions to their existence. Where did they come from? Were they always there? But the link between microbes and infectious diseases will still have to wait for a couple hundred more years in order to make that connection with microorganisms and infectious diseases. So let's go back to the miasma theory. This was the theory that bad odors were the cause of disease. Now, during the 1800s, London, like many other major cities, was experiencing an industrial revolution. Numerous factories sprouted up everywhere to help feed the demand for manufactured goods. Hundreds of people migrated from the countrysides to the cities in search of work. Thus, they crowded the cities, such as London, and overpopulated them. Now, people competed for living space and often resorted to sharing a single apartment with other families. This huge sudden boom or increase in people in such a small area led to the escalating problem of human waste. Personal hygiene was not what it is today. So compared to the smells of accumulating human waste and the lack of running water, and that of the factory smells, along with cramped living conditions, you can imagine that disease often spread rampantly with devastating results. Now, these pictures actually provide us with an example of what the cramped conditions looked like. The picture on the left shows a typical row of buildings built at the edge of the street with a sewage gutter running down the middle. Whereas the picture on the right is an example of the back of these row houses, and basically you can see that there's very little room to spare. Now, inside the houses, as I'd mentioned earlier, one family may rent a single room if they were rich enough, but otherwise they would share a room with other people, such as the kitchens, the living rooms, and laundry rooms, etc., were all in one room. Keep in mind that running water was a luxury and was basically reserved for the rich. But for everyone else, chamber pots were used. They had to be emptied on a daily basis, and where else they would empty them in the streets. Some houses actually shared an outdoor facility, such as these outhouses. These outhouses would empty into a cesspool that was dug below them, and the waste would just simply seep into the ground. Whereas other apartment houses had basement cesspools. So here's a picture of an apartment building that has 2 upper levels that were used for living quarters, with a cesspool in its basement. Now, I can imagine that the smells would often go through the floorboards into the living areas, specifically on the first floor, but would also reach the upper floors as well. Periodically, the landlord would hire a night soilsman whose job was to empty these overflowing cesspools. He would then cart the waste away to the countryside and sell it to farmers as a use of fertilizer. Now, because indoor plumbing was also a rarity, most tenant dwellers obtained their water from an everyday source, such as this well right here. So this well would actually serve people within that neighborhood. So the fact that there was a lack of strict sanitation regulations, plus the well locations, this led to a major outbreak in cholera in the Soho district in London in the 1830s and lasted for over 20 years. As in most neighborhoods of London, Soho packed its people in tightly into these living spaces near the major factories. There was a physician that lived nearby, in a neighborhood nearby. His name was John Snow, and he made it his goal to track down the mysterious cause or source of this cholera outbreak. Now again, miasma theory was the prominent theory of the time, so most physicians believed that the odor of this neighborhood, Soho, was the cause of cholera. So their belief was that if you made the area smell better, then cholera would disappear. But John Snow reasoned that in order for that theory to be true, the areas of London that generated the most toxic air would be the areas where they would have the most cases of cholera toxin. What John Snow noticed was that Soho did not smell any worse than its surrounding neighborhoods that were actually spared of the outbreak. So he hypothesized that it was something else. But what was that something else, besides this toxic air that was the cause of this outbreak of cholera? So to help him identify what was the cause of this outbreak, he went from door to door and asked anybody that would answer the door for him if there was anybody that was currently infected with cholera or who had recently died of the disease. Now, this outbreak was particularly deadly, which basically within 24 to 48 hours of contracting cholera, most people would die. Now, as John Snow went from door to door, he composed a map that you can see right here, and it's often referred to as the ghost map, since every red dot on this map represents an address where someone was affected by cholera during the time of August of 1854. Now you'll also notice in blue there are these water pumps that serve the neighboring houses. An example of this pump is shown here on the right. Now Dr. Snow's map pointed to the fact that the cases of cholera surrounding a particular pump, known as the Broad Street pump, was sort of the center of this outbreak. So Dr. Snow theorized that the outbreak was linked to a source of water, so linked to the water from this pump. Thus, he believed that the outbreak was waterborne disease, not airborne. But there were two observations that he had made early on that really didn't match his proposal. For one thing, here in green is a workhouse, which is another name for a prison. There weren't any prisoners in this prison that suffered any death or disease of cholera. Likewise, there was a local brewery where the workers there did not suffer of cholera. So Dr. Snow returned to these places and asked the people there more questions. Now he was informed that in the prison that they had their own source of water. They had their own water pump inside the prison. So therefore, if the Broad Street pump was a source of contamination, they would not have drunk from that water source. Also, the brewer workers drank beer because it was actually considered healthier than drinking the local water. So again, these abnormalities in the map actually worked to towards Dr. Snow's proposal. So Dr. Snow then presented his case to the local officials, which ultimately led to the removal of the Broad Street pump's handle. Thus, within days, numbers of new cholera cases dropped suddenly. Although many scientists and physicians still wanted to believe in the miasma theory, this was the beginning of disproving that theory. Now, it was later discovered that the Broad Street pump was actually placed within feet of an outdoor cesspool that was serviced by the residents at 40 Broad Street. It's believed that the first patient to have started this cholera outbreak was actually a young child at that address. His mother had unknowingly spread the disease by emptying the waste from her child's diaper into that cesspool, which then had cracks in it and ultimately leaked the Vibrio cholera, the bacteria that's associated or that causes the disease, which then leaked into the well and provided a source of this bacteria into the drinking water. Now still, the smells had lingered on, and the living conditions hadn't improved much, and still people wanted to believe in the miasma theory. So the city officials set out to lay sewers under the streets of London to remove the human waste and try to improve the health conditions of the people of London. Now the sewers led the wastewaters away from the city and into the Thames River, which led to other problems later on, but that's another story. So ultimately, the odors did go away and sanitary conditions did improve. Okay, so let's leave that story for a while, the miasma theory, which ultimately we'll talk about the germ theory in a second. Now, because of the discovery of microorganisms, this led to a big rush in identifying more organisms and the discipline of microbiology. So we call this the golden age of microbiology. Now, one of the scientists that contributed to this is Robert Koch. Dr. Koch was a German physician / microbiologist and is attributed as a scientist who discovered the cause of cholera as being that of the bacteria known as Vibrio cholera in 1854. Now, Dr. Koch designed a 4-step method called Koch's postulates that helped him link several diseases with specific bacteria, thus proving the germ theory. Now, this germ theory states that many diseases are caused by microorganisms. Today, we call these diseases infectious diseases. Now, the miasma theory was finally put to rest in 1910, just for your information. Now, besides Robert Koch, Louis Pasteur, a French chemist / microbiologist, also helped in the advancements of microbiology. He showed that microbes undergo fermentation as a part of their metabolism, which in turn gives food products their distinctive properties. Examples of these are yogurts, beer, and bread. Now, Dr. Pasteur also invented pasteurization, hence the name pasteurization, to kill microorganisms that are responsible for spoiling foods. Today, we also use pasteurization as a way of preventing food poisoning. So, pasteurization is a way of heating up foods for a short period of time that will kill most pathogenic organisms that may be present in food products. Now, Dr. Pasteur was also a firm believer of the germ theory. He actually came up with the hypothesis, whereas Dr. Robert Koch proved the theory. Dr. Pasteur believed that developed vaccines that was ultimately used in the treatment of rabies. So together, these two gentlemen helped advance the understanding and the practices of microbiology during this golden age of microbiology. Now, your textbook has this microbiology toolbox. I'd like to just basically zoom in on this picture here because it has some interesting terms. It talks about, obviously, microscopes. Now, we know that we need microscopes in order to observe microorganisms. What microscopes do is they magnify the images of microorganisms as well as other samples. Now, in microbiology, in order to see microorganisms, the use of microscopes alone are sometimes not enough, that we need to provide color to increase the refraction index of these organisms. So we're going to use a lot of stains and dyes within our lab. Now, in order to also grow microorganisms, we have to use certain growth media. So specifically, we're going to work a lot with bacteria. So we're going to provide them certain nutrients that they need to grow. These media could be in liquids, or they can be in solid type media that will have agar added to them. Now we'll use these plastic dishes. In the previous times, I should say, not so far away, these dishes were in glass. So either plastic or glass, we call them petri dishes. Sometimes media can be grown in test tubes. You're going to learn how to use a Bunsen burner. This Bunsen burner is a way to sterilize instruments that will be used to propagate microorganisms, to spread them from one petri dish to another, as an example. We're going to use these inoculating loops. So I'd like you to be familiar with these terms, if not for the lecture exams, but for the laboratory purposes mostly. Okay, so let's jump into another topic called classification. Now, with the discovery of many different microbes, it became necessary to classify them in the most meaningful way so as to make better sense of all the scientific data that was being collected on them, and in order to bring a better and more advanced knowledge of microbiology. So the classification of living organisms is going to help us better understand how they all relate to each other. So by organizing these organisms into different groups based on their shared characteristics. Thus, it will allow microbiologists to distinguish between the different organisms and give them names. Now, microbial classification is based on Carlos Linnaeus' early works that classified all known organisms into kingdoms. Now, Dr. Linnaeus began by placing all the known organisms of the time into 3 kingdoms. These kingdoms were the animal kingdom, the plant kingdom, and the mineral kingdom. Later on, minerals, which are not considered as living organisms, were then abandoned from this classification scheme. However, later in 1866, Ernest Haeckel, a German biologist, proposed to add 2 more kingdoms, the Protista and the Minora kingdoms. Now, the Protista consist of single-cell organisms with a nucleus. Today, we know these as the algae and the protozoa. Now, the second kingdom, the Minora, are composed of single-cell organisms that lack a nucleus. At the time, they were considered only to be bacteria. Today, we know that there is also the archaea. Okay. Now, more kingdoms were then added. In 1968, an American ecologist, Robert Whitaker, proposed that the 5th kingdom, called the fungi, would be added. Now, these fungi, at first, were thought to be as plants, so they were classified erroneously in the plant kingdom. So now we had 5 kingdoms. Now, once these kingdoms were defined or classified, they had to be joined together in a meaningful way. That's where these phylogenetic trees come in, or phylogenies. Now, a phylogeny is basically an evolutionary history of an organism. So the 5-kingdom tree of life had evolved into an evolutionary history, or a phylogeny, of all the living organisms. Those that were closely related were then grouped closer together on this phylogeny, whereas those that were not related at all to each other were placed the furthest apart from each other, and thus those in between. Now, all organisms were originally placed on this tree based on their physical similarities and their differences based on their embryology and biochemistry. So their embryology meant basically the study of how their embryos developed, and biochemistry was the study of chemical and physiochemical processes, basically their metabolism process. But then technology and our ability to analyze DNA came into play and helped us redefine this tree of life. And for this, we consider the attributes of Dr. Carl Woese. Now, Dr. Carl Woese, who was an American microbiologist, used the fact that all of these organisms in these 5 kingdoms have ribosomes. Now, he being particularly a microbiologist, he focused first on microorganisms, on bacteria. Now, all these organisms have ribosomes, and we know that ribosomes consist of ribosomal RNA as well as ribosomal protein molecules. Now, these ribosomal RNA molecules are particularly highly conserved among all species. So Dr. Carl Woese studied the gene that encoded one of these ribosomal RNAs. We call it the 16S ribosomal RNA. Because of our computers, we had evolved tremendously, he was able to analyze the nucleotide sequence of hundreds and hundreds of 16S ribosomal RNA samples that came from many different microorganisms. He compared these genes also to those of a eukaryotic organism, and this gave him the result of a 3-domain tree of life. Now, Dr. Woese had a surprise. Now, out of all the bacteria that he used in this analysis, he basically came up with 2 significantly different groups of bacteria. And so he named one of those groups as the archaea. This is where we first identified the archaea as a prokaryotic organism. But based on this genetic analysis of these three domains, our current belief is that all living organisms have evolved from a common single prokaryotic ancestor 3.5 billion years ago. As the semester progresses, we'll talk more about this. Now, the other thing that we attribute to Carlos Linnaeus, besides his beginning of the phylogeny tree, is for the development of a naming scheme. So how do we name these organisms? Now, all living organisms belong to a domain that's broken down into subcategories called kingdoms, which is then further broken down into smaller groups called phylums. These are then further broken down into classes, etc., until we get down to species. Now, Linnaeus developed the system of what we call as binomial nomenclature, such that all living organisms are going to be given a name based on their genus and species name. So for example, I have two columns here, one for humans and one for bacteria. Now, humans are classified in the domain of eukaryotes. They are then further broken down or found in the kingdom animals, all the way down to genus Homo and species Sapien. Thus, our binomial nomenclature for humans is Homo sapiens. Now, we can do the same for every single living organism. For example, one of the bacteria that survives in our gut, the Escherichia coli, most of us know it as E. coli. This E. coli is found in the domain bacteria, also has the same kingdom bacteria. The phylum is the Proteobacteria, subcategorized in the class of Gamma Proteobacteria, etc., until we get to its genus Escherichia and its species coli. So its binomial nomenclature name is Escherichia coli. Now, this binomial nomenclature was established in 1735 and still used today. Originally, all known organisms were given the Latin-based name, but more recently, new names have different origins. For instance, these names can have a Greek or an English background. Now, each living organism was and still is given two names. As I had mentioned, the genus as its first name and its species as its second name. Now, it's important that you know how to write these organisms because you're going to find this in scientific literature, and scientists are very particular on how we write these names so that we can be consistent. So whenever we refer to a genus of an organism, that genus's name will be written with a capitalized letter. So here I have genus with a capital G. As an example for Escherichia coli, Escherichia is always capitalized. Whereas the species is written in lowercase, it's never capitalized. So coli it never has a capital C. Now, when you're writing scientific papers, you're going to rewrite the organism's name over and over again, so it's customary to abbreviate these names. So the first time you write the organism's name, Escherichia coli, you're going to write it in full length. But the next time you write it, you're going to abbreviate that name, whereas the genus will just have the first letter with a period. Now, in either way, whether you write it the first time or in abbreviated form, the genus and species should either be italicized if you're typing it or underlined if you're handwriting it. So these are the rules of writing the name of species. Okay, so during this semester, you're going to be introduced to the many different types of microorganisms. They're going to be grouped into three broad categories as I've shown here: the eukaryotic microorganisms, the prokaryotic microorganisms, and the acellular microorganisms. And you're going to need to know which group that these will follow in. So to begin with, you're going to learn about protists, and protists are going to be a combination of the algae and protozoa. These are going to be polyphyletic. They're going to be very difficult to separate them phylogenetically. We're still trying to get on how we're going to classify these organisms, but they are distinct. For instance, the algae are going to be single-cell or multicellular organisms. Most of them are going to undergo photosynthesis. Whereas the protozoas, they are all single cell, and unlike the algae, they don't have a cell wall. So and they're not going to do photosynthesis. So there are differences between the two groups. This is one of the algae called diatoms. They're kind of neat. Another type of algae that you'll be introduced to are these dinoflagellates. The next eukaryotic microorganisms are going to be the fungi. I've also already mentioned a couple of them today in the form of yeast. These are yeast that produce our food products. You'll learn about mushrooms and bread mold. They're also fungi. Now, like mushrooms, these are macroscopic multicellular, but they're closely related to the microscopic mold and yeast cells. So that's why they're grouped together as fungi, and that's why we study them in microbiology. Now, these guys were once classified as plants, but you're going to find out that they are very different from plants. For one, they don't do photosynthesis, and their cell walls do not have cellulose, but they have chitin instead. So there's a lot of differences. Now, helminths I tend to skip over because I don't know much about them. These are roundworms and flatworms. So for this semester, you're not going to hear about helminths. We're then going to jump to the prokaryotic microorganisms, the bacteria and the archaea. Now, the bacteria are single celled, just like the archaea. They're found in every habitat on Earth. They will be the most diverse microorganisms that we're going to learn about, and some of them are going to be human pathogens. Now, the archaea, there are none of them that are known as human pathogens because they live in such diverse environments. Just like the bacteria, they are prokaryotic. They don't have a nucleus. They're a single cell, but there's some differences that we're going to learn. Now, here is the thermal vents found in Yellowstone National Park, and these waters can reach up to 200 degrees Fahrenheit, highly boiling water. There are these archaea that will live down deep in that water. Some of that water is also very acid condition. Just to give you an example of an extreme condition. So you and I could not survive under those conditions, but these archaea microorganisms can. Lastly, towards the end part of the semester, we'll look at viruses, prions, and viroids. Now, these are a problem in trying to prevent infection from these organisms because they are somewhat they hijack the host cell, such as human cells. Now, viruses, they're not going to be true cells. They're basically a piece of genetic material that's either DNA or RNA that's protected by a protein coat. They're not considered to be living. Prions, on the other hand, are even more simple than that. They're a single protein. And some of these proteins can actually be found in humans. Also, the last acellular are these viroids. Now, these are not human pathogens, but they're plant pathogens, and they're just a single RNA molecule. So we're going to learn about these as we go along. So as the semester progresses, you're going to get more information. Now, just to give you an idea that microbiology is a huge umbrella of different branches of studies. And just to give you an example of these studies, I'm a bacteriologist by trade. I study bacteria. Whereas there are other branches such as mycology that studies fungi. There's virology that studies viruses. There are those that study just protozoas or algae. These are parasitology or protozoology. And then there are those that study how the host reacts to these infections, to pathogens. These are called immunologists. They study immunology. So just to give you an example of the different branches. So that's going to be it for the introduction to microbiology. As the semester progresses, as I promised, we're going to delve deeper into these basic ideas.